Well, first of all, I want to thank the organizer for giving me the opportunity to talk about my work. So my talk is going to be slightly different from the one that you've heard before, because mine is a very little story about how we've been exploiting the Cas9 system to improve or trying to improve what is a challenge in differentiating pluripotent stem cell. So we know that the advent of pluripotent stem cells such as iPS or ES opened the cell therapy field. So in theory, those cells could be used to produce any cell type of the body, and this could be then transplanted for different kind of disease. But what we know from the culture, from the differentiation, is that it's not easy as it seems to be, and that's where the Cas9 could really help us facing this issue. So I'm talking today about the application of the dead Cas9, so I'm not going through any genome editing application, and that's what we want to do. So we want to change the cell fate by activating endogenous gene expression. So what people have been successfully doing since a while ago is that if you take a cell and you put specific transcriptional factor, which are proteins that activate the expression of a gene, you can change the cell fate of this cell. So this is the first example reported in which fibroblasts have been transdifferentiated into muscle cells just with the use of one transcriptional factor. Those are other successful experiments in which people have been adding this DNA to the cells and transform a cell type to a completely different cell type. So all this system relies on providing the cells the gene. So you just take a cell and you overexpress the gene in those cells, and you force the cells from going to one cell type to another cell type. But all those cells actually already have the copy of the gene in their genome, because these are human genes or any other kind of genes. So that's where the Cas9 really made the difference, because we know, well, these are the wild type Cas9. But if you take this Cas9 and you mutate the catalytic site of the protein, what you end up with is what is called a dead Cas9. So it's a protein that still is able to localize into the nuclei, to the gene of interest, but it's not going to cut and it's not going to introduce any effect on the sequence of the gene. But what you can do is that if you fuse this protein with a functional domain, you can actually change the epigenetics at that locus. So you can mediate activation of the gene, you can mediate repression of the gene. And if we take, for example, this situation, if we have a cell fate A in which the gene X is not expressed, what you can do is that you can drive your Cas9 to the locus and use a Cas9 with an activation domain fused to, and you can reactivate the expression of those genes. So you don't provide the cells the sequence, you just say to the cells, express the gene that you already have in there, and that's the same for repression. Is this going to work? Yes, because recently there's been paper showing that you can change the cell fate with the use of the Cas9. So this was the first one where they did exactly what people have been doing in the first transdifferentiation experiment, but in this case there was no exogenous cDNA, no exogenous DNA provided to the cells. They just activate the expression of MyoD into fibroblasts, so their own gene in their own genome, and those cells were successfully transdifferentiated to myotube. Has been reported also the transdifferentiation of fibroblasts into neuron with the activation of 3 different genes. So that's really showed us that we can do this with the use of the Cas9, because one of the concerns was that usually people have been used to think that you needed a lot of gene, like the high level of expression of the gene to change the cell fate from A to B. But this and the Cas9 activation showed that sometimes it's challenging to get like very high level of expression. So that was was a concern in the field, but those papers really show that you can do that. So, and here it comes my challenge. So our lab is focused on the production of cells, blood cells, from iPS cells, and we are particularly focusing on red blood cells, which are our erythrocytes. Those are extremely differentiated cells which lose their nuclei and they express massive amount of globin, because their goal is to transport the oxygen and remove the monoxide from your blood. So what we know is that if you take adult progenitor cells, adult progenitor stem cells from different sources, such as bone marrow or cord blood. You put them in vitro, you differentiate them to red cells, and it works. You can get red cells, which are like adult-like red cells, fully nucleated, perfectly nicely cells. But if you do this from human pluripotent stem cell, like iPS or ES, you generate what is a progenitor cells in culture, and what you get is this. So you get nucleated red blood cells. And those cells are very much like the red cells that are in the embryo. So because early in the embryo, your red cells were not nucleated, were like those one. So we said, okay, so what's the difference between why our system is not working? We know that if you take progenitor, you put them in culture, it works. So it's not the in vitro culture condition. So it must be something between those cells that is different. So what we did is to compare the transcriptome of those cells. So we generated a microarray to compare their expression, and we came up with a list of different genes that are expressed at different levels, specifically transcriptional factor, which are the molecule that drives really the cell fate. And so we said, okay, people have been trying to reprogram those cells to adult-like progenitor cells using exogenous DNA, as I showed you before. We said, okay, let's try to do this with the Cas9. So what we want to do is take an in vitro produced cells and then activate the expression of those genes that are missing and try to make those cells look like adult progenitor cells. So different functional domains have been used to activate the expression of the locus. These are some examples of them. So people have been fusing those domains to the Cas9, deliver them to the DNA and activate the expression of the gene. This one seems to be the more effective, which is based on synergistic activation of the system. So you have your Cas9, VP64, which is a viral protein that activates the transcription. And then you take the advantage of using aptamer, your guide RNA, which can bind then this fusion protein, because this little part of the protein is going to interact with the aptamer. So what you do is that you kind of amplify the activation of your gene of interest, mediated by synergy of those molecules. And the great advantage of this system is that while before to activate the expression of a gene, you needed lots of different guides. So you would have like kind of many molecules of Cas9 to activate the gene. In this case, you need only one. So if you want to activate a lot of gene, you just need to pick one guide for each gene, and you can mediate multiple activation. And another important thing is that all this system is based on the TSS. The TSS is the region where the transcription starts, and this information is available for all the genes in your genome, while other systems such as P300 are based on an answer, which are specific functional parts of the DNA which are described for some gene but not for all of them. So let's say if we need to activate a gene and we don't have this information, we cannot do. With this system, we can activate every gene of the genome. So, and here comes what I did. So that's a system that is available so far. So for those who are not familiar with plasmid, that's just simply to say that this system was based on the use of 3 different pieces of DNA. And because you needed all of them in the same cell to mediate the activation, you had to put your cells under a triple antibiotic resistance that for differentiation is a real big challenge. And deliver all of them in the same cells is going to be like another challenge. So what I've been doing so far is generate a system that contains all these different parts in the same plasmid. So a very simple system all in one, which would completely eliminate the heterogeneity in the population. And then we also added a fluorescent tag. So if you have to do a differentiation, you want to pick only the cells that successfully express your system, you just take those cells out of the culture. And then that's just to say that we used a system that could be used to express the system for a while, so in transient, and can be integrated into the genome, so you could express it in a longer time frame. And that's basically the system that we developed. So we have our dead Cas9 fused to an activation domain. This is the protein that synergistically activate the locus, and that's our fluorescent tag. And this is our guide RNA. We need one for every gene. So this plasmid, basically, if you make one plasmid, you can activate your gene of interest. And as it was said before, it's extremely easy and it's extremely cheap. So you pick your gene of interest, and in 3 days you've got your plasmid. You can test it, activate, and test for phenotype in your differentiation. So what we did first was to check if our system was really able to activate the endogenous gene expression at the locus. And to do this, we use cell lines that are easy to transfect. So these are not embryonic stem cell lines. These are other cell lines which are really easy to transfect. And as you can see here, by the expression of our tag, the cells were happily expressing the cassette. And that's the activation that we achieved. So these are mainly transcriptional factor involved in the hematopoietic system. And as you can see here, we got like very nice induction of endogenous gene activation. Some of them, though, showed resistance to the activation. And we wanted to understand why some of the genes were easy to activate and some of the genes were not. So what we did was to correlate this with the basal expression of gene, because we know that every cell line has a different expression level for every gene, and that's what's the case. So it's really easy to activate the expression of a gene which is not expressed in the cells, but then if you take a gene that is already expressed, you cannot express it at higher level. And that was also like an indication that probably this system would be like a more physiological system to activate gene expression, because if you overexpress a gene, you just load the cells with a massive amount of transcription factor, which would never happen normally in a cell. So it's a very different system. While in here, it seems that probably what we were doing was something like pseudo-physiological activation of the gene. And we went on to study this at single cell level. So what we did was to target our gene of interest, one of our main interests. And as you can see, compared with the empty vector where you don't have the guide RNA, so you cannot localize the Cas9 in your gene of interest, you don't see many active cells. And upon addition of the guide RNA, you can see how the different cells show expression of the target now. So we use a system available where you can screen for positive and negative cells. We quantify this, and we noticed that there was also variability not only between the different genes, but also in the same gene. So some guide seems to be more efficient, some guide seems to be less efficient. And at this stage, we wanted really to compare this with the overexpression which was available before, was the old system to have a cell express what you want. And that's what we got. So as you can see here, the cells express a massively high amount of protein when you provide them the sequence. And if you compare this with our system mediated a level of expression which seems to be comparable to the control and was significantly lower than overexpression system. Then we went on and we wanted to check also if, okay, you get the expression of your protein, is then the protein functional somehow? So we picked a protein that after being expressed has to go to the membrane of the cells. And in this experiment, we also wanted to test if the expression was really physiological. So we picked one cell line which expressed the gene and one cell line that do not express the gene, activate the expression of this gene. And as you can see, this is the cell line that already expressed our target gene. So you can see in here, these are positive cells above the line. And in here, you can see that before the addition of the guide RNA, we don't get positive cells. If you add the guide RNA, we get positive cells. And those proteins seems to be functional because they can migrate to the membrane. This is just the quantification of it. And again, we compare the expression level. And as you can see, this is our cell line where we added the guide RNA. We activate the gene, but we don't see like a massive upregulation of the gene, which is good. And then we moved on to our UNIS. So what I want to say is that a lot of people have been using cell lines which are easy to manipulate. UNIS are not easy to manipulate, and it's really difficult sometimes to find the balance between manipulation and cell viability. So that was really the main point of our work. So to do this, we used reporter line, which means that you can simply put like a fluorescent protein in your gene of interest. And then if your cells turn fluorescent, it means that your gene is active. If your cells do not turn fluorescent, you didn't activate the expression. So this was a great genomic tool to test our system, because we could look at cells that were red, because this was the tag of our of our plasmid, and then we could test also GFP, which is a green fluorescent protein, looking for green cells that were activated. And we could see that we could mediate the presence of GFP positive cells, so activated cells above this line. As you can see, again, they look different. Different guide mediate different activation level. That's the quantification of this. And also within each cells, you could get also different level of expression, which is really good because sometimes in some system, also the relative expression of different transcription factor decide one cell type upon the other one. So with this, you can really tune what you want to get. So let's say we need a cell fate which is driven by this level of expression, then you pick this guide. You want more, you pick the other one. And finally, because the system that we use was just the all-in-one version, the one available before, so people have been asking, is it really worth to go for your system, or is it better to use the old one? And that's where you can see the difference. So these are yes, and as I told you, it's not really easy to work with embryonic stem cell. And you can see how with our system we got way more live cells. You can see it in here as well. This is a picture of the plate. You can see very few cells, and their nuclei is in blue in here. With our system, we get way more cells. And that's true also upon time. So you can see that our cells are quite happy and proliferate, while the other cells just die. And with this, I can conclude, and I can tell you that we develop a system that successfully activates the expression of endogenous gene. This seems to be related to different things in the cell, such as the basal expression and also the guide RNA that you pick, and that this system showed much higher viability compared with the system available so far, which is good if you want to generate nice and happy cells. And that's our future plan. So now that we have a system in our hand, what we want to do is to manipulate the differentiation of hematopoietic cells in vitro, try to make the right cell type, and then we are going to test this with different hematopoietic tests that are available so far. And with this, I want to thank the people that really helped this work to become real, and all the people at the screen, which has been like helpful all the way through my postdoc. And that's it.
Okay. Any question? No? Okay.
Okay.
I have a
Okay, good.
What about the blood type?
Oh.
Yeah, I mean...
You mean which type are you going to make?
Yes, exactly.
Okay, so the thing is that if you can make the stem cell, the hematopoietic stem cell, you can make any cell type. So that's the golden cells everyone is trying to make in vitro. Then what you can do once you have that, you can manipulate and let it become a red cells, or you can go directly for red cells. So the advantage of manipulating the transcription factor is that you can jump in theory as you want. So you just compare the transcription factors, the two cell types, and you jump to red cells, you jump to macrophage, you jump to neutrophils. But if you get the stem cell, you're done, because then from the stem cell you make all the cell type.
What's the therapeutic pathway that you envision towards application? Will you have like a bank of iPS cells or ES cells for, let's say, I think you were talking about blood types, so type A, type B, or will you just make them all O negative?
Yeah, so the strategy that we use so far is to use an iPS live derived by O negative. So this is the way we are going now. So if you generate a stem cell which is O negative, then you could make blood cells that could be used for transfusion in any patient really. But then if you want to use, and that's also like a big advantage of using red cells because they lose their nuclei. So if you also include the transgene, then your cells will not have it anymore. But then it's going to be different in case of nucleated cells. For example, if you want to make macrophage, in theory in the future, maybe we're going to have our own cell bank and make our own macrophage from iPS. But I guess we are a bit far from this.
So your system of transcription, I think it was lentiviral, right?
No, everything is free. So everything is virus free. Everything is transfected.
And then is it, so you get like constitutive expression or is transcript?
You can do both because that's the advantage of the system. You can do, if you just transfect your piggyback, is expressed for a certain time that depends on the speed which your cell cycle, because it gets diluted upon cell cycle. But you can also put transposase, which are proteins that integrate this into the nuclei, so then you can have it expressed stably. And you can also remove it, because the advantage of the piggyback, and that's why people have been using for therapy, is that they are scar-free. So you put them in the genome, and then you put the transposase again, and you remove them, and that's absolutely like the genome is like as it was before.
Okay. Any other question? Okay, now is the lunch outside, and this was the poster session, so the authors of posters should be close to the posters, okay? And let's thank again Antonella for